Okay, so starting with this video where we left off last time, we will begin with this, and then we will move on with today's lesson. So we finished up about here. We didn't have time to watch the video, and we will continue with this and move on with the nephron and how the nephron filtrates and reabsorbs things.
The kidneys are organs of the urinary system. They regulate the water content of the body and remove metabolic waste through the formation of urine. How does urine form? When blood reaches the glomerulus, it is maintained at a relatively high pressure. The high blood pressure forces water and small soluble molecules, such as glucose and amino acids in the plasma, through the thin walls of the glomerulus and the Bowman's capsule into the capsular space. This process is called ultrafiltration. The fluid that is filtered into the Bowman's capsule is called glomerular filtrate. The filtrate has a similar composition to plasma. However, no plasma proteins and blood cells are found in the glomerular filtrate because they are too large to pass through the walls of the glomerulus and the Bowman's capsule. After ultrafiltration, the glomerular filtrate flows through the kidney tubule. In the kidney tubule, useful substances in the filtrate are absorbed back into the surrounding capillaries so that they are not lost from the body. This process is called reabsorption. It occurs mainly in the proximal convoluted tubule. In the proximal convoluted tubule, all glucose, amino acids, and most salt are reabsorbed by diffusion and active transport. As many solute molecules are removed from the filtrate, the water potential of the filtrate rises. Therefore, water in the filtrate enters the blood by osmosis. The reabsorption of water raises the concentration of urea in the filtrate. Thus, urea in the filtrate diffuses into the blood. Further reabsorption of substances takes place in the remaining part of the kidney tubule. After reabsorption, the remaining filtrate in the collecting duct is called urine, which will be passed out of the body when we urinate.
So that's just a summary of the whole process. And don't start again. Don't start again. No, don't start again. There you go. Okay. But we're going to move on with today's material, talking about the nephron and the actual filtration process. So this is kind of the real meat of what's going on within the kidney and how the process works. So the filtration process is starting in the nephron, looking specifically at the glomerulus, and then as it goes through the parts of the tubule, then it's going to be absorbed and then reabsorbed, and materials get pushed out, and different parts of the descending loop and then ascending loop are going to take materials out and then put it back into the bloodstream. So as it's dipping down into the medullary, it's going to take some things out and some things put back in, and it's really kind of complex at which stages these things occur. So a lot of this I put in Google Classroom, and this again is like the main part of what is going on within the kidney and how, and whatever is left over within the duct, that is what is staying behind and being made into urine. So again, remember the afferent arteriole is larger. Afferent arteriole is larger, and what is going out is the efferent arteriole, and that is smaller. So 80% of the blood plasma stays, and only 20% is filtered out into the tubules, and then we have a lot of filtration that's going to take place. So the plasma volume entering the afferent arteriole is 100%, 80% stays, 20% is filtered out, and then 19% gets reabsorbed back into the blood. So here is our filtration. About 19% gets reabsorbed back into the blood throughout the process, and 99% of the plasma entering the kidney returns to the circulatory system, and only about 1% of the volume is excreted as urine from the body. Now this chart is in Google Classroom, and this chart is going to determine all of the different materials that get excreted, all of the different ions like potassium and sodium and magnesium and phosphorus, and all of that will get excreted along with water, and the varying amounts and where they get excreted and reabsorbed, because each part of the nephron will be responsible for absorbing or excreting different ions. So I put a lot of these pictures in Google Classroom because they all have different benefits, and they all have different descriptions, and they do a good job of explaining the process. What's important to pay attention in these pictures is the arrows. Some arrows are pointing into the ducts, and anything that's going into the ducts is going to be collected as urine and excreted from the body. And anything that's pointing out, like this, is going into the blood. So whatever's left in these ducts is going to be urine, but whatever's coming out is being returned to the blood. So in these ducts is urine. Anything outside here, this is all going to be returned to the blood. And then these do a nice job of saying step by step, and a lot of this is color coded too. So water is color coded blue, and then this one's glucose, and so on. So the color coded ones make it a little easier to understand that here in these collecting ducts, the collecting ducts is the last step, and several nephrons are going to empty into one collecting duct. So like this would be where a nephron is, this one's a nephron, and so on. The water will be absorbed out of these collecting ducts and going back into the blood. Now the nephrons, the kidneys, they're the main area that is going to regulate the water balance in our bodies. So we've noticed this before, I'm sure everybody's noticed this. If you drink a lot of water, you're going to urinate a lot more, and your urine is going to be more of a pale color. Whereas if you're dehydrated, then you're not going to urinate as much, and your urine is going to be more of a darker yellow color. So this is all part of the process. It's going to be how your body just regulates the coloration, and how your body regulates the process of water absorption and water balance. So a couple things to keep in mind. The sciencey words is secreted and reabsorbed. When we say secreted, that means that it is going into urine formation. When we say reabsorbed, that means that it's going back into the blood. Your body is reabsorbing it back into the blood. But when we say it's secreted back into the collecting duct, the tubules, and that's going to be made into urine. So as we go through these notes today, just keep in mind that secreted means urine, reabsorbed means that it's being reabsorbed back into the blood. So I tried to keep that consistent throughout the notes. Because at this point, everything has been pushed out into the tubules. And so we are reabsorbing stuff into the blood, but the blood will also secrete some other things that we still don't need back into the ducts. Alright, so water, 99% is reabsorbed, and this is kind of in general. So remember that this is in the ducts itself after the glomerulus, after the Bowman's capsule, after we've squeezed everything out, because we squeeze out the good and the bad, and now our body's like, okay, we've got everything out, now let's take care of what we need and get everything we need back. So 99% of water is reabsorbed. And then about 99.5% of sodium or salt is reabsorbed. Urea, however, only about 5% or 50% of that is reabsorbed. And phenol, none of that is reabsorbed. Our body wants to get rid of the phenol. There are 3 processes that our body uses. There's the passive diffusion, which doesn't use any energy. And then there's 2 types of active transport. There's a primary active transport, which uses ATP. That's the one we learned about before, which is just using ATP actively to push things across the membrane. But there's also something called secondary active transport. We didn't really talk about this one before. It does not use ATP directly. Instead, what it's doing, it's kind of like taking active transport and then having another one kind of piggyback and kind of follow it. So you're using active transport, but another one kind of hitches a ride and kind of follows along behind the other one. So like you have sodium that's going through active transport, and then another one is going to follow along right behind sodium and kind of hitch a ride with sodium, and that's secondary active transport. So sugars and amino acids are going to do the secondary active transport. So there's 2 pathways of absorption. There is the transcellular pathway, which goes across the cell itself, and then there's what's called the paracellular pathway, which is between the cells. So sometimes it's going to go, and then this is the blood. It can go across the cell, and then sometimes it goes between the cells. So these are the two different ways it can get reabsorbed. It depends on what the material is, which pathway it's going to take, whether it's going to go between the cells or go through the cell. So. And this kind of shows how the process works. This is primary active transport. We have the diffusion across, so this is like a cell, and this would be the blood over here. And then we have our active transport, which needs energy. So this would be the across the cell. And then we can have some that would go between the cells here. But again, if we have active transport that is taking place where energy is needed, then this would be the process of using that ATP to push materials actively across that. And this is in Google Classroom. But this would be secondary active transport, where we have it kind of piggybacking. So this is glucose, and we have the sodium moving across, but we have glucose kind of catching a ride with it. And this time we can have it going, this is counter transport, so we have sodium going this way, and as the sodium goes this way, the hydrogen is moving the opposite way. So sometimes they can go both directions in the same direction, and then sometimes they go opposite each other. So sodium goes one way, hydrogen goes the other. So this would be like secondary active transport when they're both going the same direction and then they're going opposite directions. All right, so this is an important slide just because it shows kind of all in one place. This is reabsorbed, and this is what is going into the blood, and this is what is going into the tube to make urine. So this is what is being made into urine, and this is what's going back into the blood. So what is going into the blood is all the glucose and amino acids. That's what our body wants back. It wants the glucose back, and it wants the amino acids back. It wants most of the sodium, 50% of the chloride, and about 85% of the bicarbonate. Most of the water gets back, and about 50% of the urea and all of the potassium gets back. But as far as what gets into the urine, this is kind of variable. It depends on what our body needs at the time. So think of like kind of electrolytes, the acid-base regulation, and also hydrogen ions. So that is going to vary what our body actually needs. Hydrogen ions is usually what is monitors the acid-base regulation here. So there's going to be some variation with that. one is showing how this happens across the membrane as well. We've got, this is the proximal convoluted tubule and all of the transportation that takes place across the proximal convoluted tubule. There's a lot of absorption that takes place in this particular area. So we have sodium crossing here with active transport, and this is potassium that's going one way. So the sodium-potassium pump has a lot of action taking place. Sodium is going one way, and then also bicarbonate, and then we have chloride going the opposite direction. Calcium is also something that's being reabsorbed as well in this area. So even though it can look a little bit confusing if you break it down and look at it piece by piece, in this case energy is being used in the sodium going one way, potassium going the other, and this one sodium and bicarbonate and sodium, and then the calcium is here in the middle. So PCT, proximal convoluted tubule, that's what it stands for, so just an abbreviation so you don't have to write it out all the time. Sodium, potassium, and ATPase. Remember, anything that ends in ACE is an enzyme. Sodium and potassium are being reabsorbed in chloride and water, and this is all in the first half of the proximal convoluted tubule. Sodium is reabsorbed by co-transport, and that's where it's kind of doing that piggyback part with the secondary active transport. So it's getting moved through that proximal, so it's almost as soon as it comes out of the glomerulus. So our body wants to get that sodium and that chloride as quickly as possible, and it wants to get that glucose and those amino acids as quickly as possible. So it's trying to get it all back in the blood very quickly. It doesn't want it to have to go through the rest of the nephron. Chloride is not going to be reabsorbed in this area. So as we go through this, we're basically looking at each section of the nephron, and we're going to kind of follow the nephron through this part. And so at the proximal tube, this is what's going to be absorbed. At the loop of Henle, this is what's going to be absorbed, and so on. So then you get to the second part of the proximal tube, and sodium gets reabsorbed mainly with the chloride ions. So sodium and chloride together, which is basically salt. And the higher the chloride concentration favors the diffusion, so sodium is passively reabsorbed. So here you can kind of see how that's working. The sodium and the chloride are going together. Now, if you have a really salty concentration, the water is going to naturally follow this. So you're soon going to see that water is also going to be reabsorbed. The salt is going to naturally, the water is going to naturally follow the salty content. So that's going to favor osmosis. And so you don't have to write this down because this is more of an anatomy thing, but it shows the parts of the nephron. So we've got the proximal convoluted tubule, and then the loop of Henle is actually divided into a couple parts. There's the thicker descending, and then there's the thin descending, and then we actually have the loop of Henle, and then we have a thin ascending, and then we have a thicker ascending, and then the thick and the thin are going to allow for different absorptions of different materials. So that's why you don't have to write it down. It just, the visualizing of how it is divided allows for you to kind of understand why different things get absorbed in different places. So again, that's more of an anatomy thing, but just kind of helping you visualize that there is this whole thing is called the loop of Henle, but there's a thicker part and there's a thinner part, and then it dips down. So this is the descending limb, this is the actual loop ascending limb, and then there's a thicker part, and then this is the distal convoluted tubule over here, and this is all the collecting duct. So there's a high permeability to water, and this is in the descending loop of Henle, this part right here, that's highlighted. High permeability to water and moderately permeable to most solutes. So it is really thin, so water is going to be able to flow freely out. The thinness is shown here in this cross section. And moderately permeable to most solutes, so the other materials can usually flow out pretty freely. It does have few mitochondria, so little to no active reabsorption. So those active transports that needed ATP, that's not really going to happen so much here. There might be a little bit of it, but because mitochondria are necessary for ATP formation, we're not going to see as much of that take place in the descending loop of Henle. So the thick ascending limb reabsorbs about 25% of sodium chloride and potassium, absorbs a large amount of calcium bicarbonate and magnesium, and secretes the hydrogen ions. So this part right here is much thicker, as you can see in this cross section. The hydrogen ions are coming into the tube, and the potassium, chloride, sodium, all of that is going out and being reabsorbed back into the blood. And that's why that secretion and reabsorption gets a little weird when you learn those words. Hydrogen ions are coming into the tube, and the sodium and all of that is being reabsorbed back into the blood. That's what that means.
Is the restroom quick?
Mm-hmm.
Like, how's your slides looking like? We'll worry about finals.
Is what?
You're not running the Bulldog Watch.
Is paper?
Yep.
Is presentation?
Well, I mean, I have 4 finals due this Friday, so. With the boring
Are they like online or something?
Yes. So
When's the final for your class?
Hmm?
Seriously.
What?
When's the final for your class?
Finals for dual credit aren't due quite as soon as some of the other finals. So I have to have grades for seniors due by the 10th. So this is sodium chloride and potassium in the thick ascending limb. They move by paracellular diffusion. It goes across between the different cells. So here are the tubes, or the cells within the ascending limb. And then it can go, the sodium, potassium, magnesium, calcium kind of squeezes between the cells. ATP for the sodium and potassium pump. And then we see that sodium and hydrogen can kind of piggyback. That's that secondary transport we were talking about. But a lot of them are just going to diffuse across between the cells here. And this is in Google Classroom. So glucose is a big deal. Glucose is needed for cellular respiration. And so glucose is reabsorbed along with sodium, and this happens in the proximal tube, and this happens by secondary active transport. Glucose is a pretty big molecule. So in order for it to get reabsorbed, it's going to need a little bit of help. And that's why secondary active transport is going to be necessary. It can't just go across the membranes by itself. It's going to need something to help it along. And the glucose is going to use that sodium to kind of help it push it along and help piggyback it. So essentially all the glucose is going to be reabsorbed. Our body doesn't want to lose glucose. If you if you ever go and they ask for a urine sample and it shows that you have sugar in your urine or glucose in your urine, that's never really a good sign. Your body should be reabsorbing all of the glucose from your body. If it is leaving any glucose in the urine, then that is not something your body should be doing. There's something wrong with your kidneys if glucose is being left behind.
What was number one? I don't need the whole slide, I just want to write down what number one was. I already have it, it's this far, never mind.
Yeah, it's back pretty far. So, the proximal tubule here is sodium and then glucose that's kind of piggybacking off of it. And then we have the glucose here and the sodium here. And it's gotten into the cell, and now it's using the sodium potassium pump, the glucose being reabsorbed and then the sodium, potassium over here pumping it as well. So it's a secondary type of transport because it has to kind of join it in order to get where it needs to go. Alright, hydrogen in bicarbonate, the acid-base balance of our body, balancing that is also an important part of the nephron and the kidneys. And then hydrogen, of course, is part of the pH scale. I mean, it's what creates the pH scale. And so it's going to adjust the levels in our body. And so how it's secreted or how it's absorbed is going to determine the pH levels, and we want to keep our blood pH level as close to neutral as possible. So absorbing or secreting hydrogen ions is going to help to determine that. This is also done through secondary active transport. The proximal tubules, the loop of Henle, and the early distal tubules are going to be where the hydrogen ions are secreted. More than 90% of bicarbonate is reabsorbed passively in this manner. So bicarbonate is more of a buffer, and it's going to help to neutralize the acid if the acid gets too strong. So our body tries to reabsorb this back into the blood to help neutralize the acid if the acid gets too strong. So primary active transport of hydrogens begins in the distal tubules. So now we're past the loop of Henle, and now we're to the distal convoluted tubules. And lots of hydrogen ions in bicarbonate can be transported in these distal tubules. And they're transported directly by specific proteins and hydrogen transport ATPase. And all of that can take place within these distal tubules by active transport.
Just something else to do. That was close next.
one that's on the outside. And then you put that at the bottom. And then you just foil the top and bottom, basically.
I don't know what to do with the extra numbers in the front. That's 4 and 2.
So you put the 4 on the part of the P this time.
This shows how you are actively transporting the hydrogen and the sodium, and also the sodium and the hydrogen here. It can be moved back and forth, and then this is the bicarbonate to help balance that acid base as our body needs it. So potassium does get reabsorbed and secreted as our body needs it. It gets reabsorbed in the. You guys taking notes? Potassium gets reabsorbed in the proximal convoluted tubules and the ascending loop of Henle, and gets secreted in the distal convoluted tubules. Because this is a complicated topic, and you can bet there's going to be a question that's worth a lot of points on the final about this. This is one of those topics that's a little complicated, and it may not be the most exciting, but you can bet I'm going to have a question about absorption and secretion of the nephron on the final, because it's important. And yes, it may not be exciting, and yes, it may not be a little boring, but it's important. And just because I know you guys aren't paying good attention today, I'm definitely going to make it worth a lot of points on the final.
When you type in Henle, it says it's not a word.
Yeah. Alright, we will stop at this slide though. We'll pick back up here tomorrow. This is a good place to stop.
Oh, hold it, we're gonna break it.